Hello everyone, welcome to another video of basic concepts of genetics. In today's video, we will discuss about multiple alleles. In the previous videos, we have discussed about Mendel's law of inheritance, where we have discussed about Mendel's law of dominance, law of segregation and law of independent assortment. Now, in each of the 7 pairs characteristics studied by Mendel, there were only two alternative forms of each character. That is, there were basically two contrasting characters present in the gene, or there are two kind of alleles present. Now, what is meant by multiple allele? Multiple allele is nothing but when there is expression of or the presence of more than two alleles in a single gene, then those alleles are known as multiple alleles. Now, multiple alleles are always present in a population or for a particular species, but a single person or a single human being cannot have multiple allele. He can have two combinations of the alleles present, as humans are diploid in nature. So we will have to keep in mind that a single person can only have two alleles for each gene and multiple alleles present in a whole population. So what are the examples of multiple allele? One of the classical example of multiple allele is the ABO blood group system in humans, which generally consist of three kinds of alleles, and upon those three kinds of alleles one individual can have the types of blood groups present, that is A, B, O and AB. There are also other kind of examples such as the rhesus blood group in humans, expression of wings of Drosophila, coat color in rabbit. So now let's discuss about the ABO blood type in humans and the alleles present in it. There are basically three kinds of alleles present for the expression of the blood type in humans. The number one is capital IA, the second one is capital IB and the third one is small I. The capital IA encodes A antigen, the capital IB encodes B antigen, the small I encodes no antigen. So now we will have to keep in mind that on the basis of these three alleles, there is a combination between any two of these three alleles, and the expression of the blood group takes place on the basis of those two alleles, where the capital IA and capital IB are dominant alleles, and when both of them are present, then there is a presence of codominance. So on the basis of that, on the basis of dominance, there is a relationship between capital IA and capital IB, where capital IA equals to capital IB and small I is always recessive in nature. That is, if somehow there is presence of capital IA and small I, then the expression of the blood group will be capital A, that is A blood group. Whenever there is presence of capital IB and small I, then the expression of the blood group will be B blood group. That is, I is always recessive in nature, and only if two small I is present, then only the expression of the blood group will be O. So now let's talk about human blood group system. There are basically four types of phenotypes present, that is A, B, AB and O, where we have said that A is dominant in nature, B is also dominant in nature. When there is expression of AB, it is codominant in nature, and O is recessive in nature. That is, when there is presence of two small I only, then only the O blood group will be expressed. So on the basis of that, there are 6 kinds of genotypes present on the basis of the 4 type of phenotypic ratio. So when there is a blood group, then there can be two possible combinations. One is capital IA, which is homozygous in nature, and the next one is capital IA and small I, which is heterozygous in nature. Again, for B blood group, there can be two type of combinations, where capital IB is homozygous in nature and capital I and small I is heterozygous in nature. For AB blood group, there will be presence of both capital IA and capital IB, which is heterozygous in nature. And only for expression of blood group O, there will be presence of two small I, which is homozygous in nature. So now let's talk about the antigen present in each of the blood type. A blood group consists of A antigen, B blood group consists of B antigen, AB blood group consists of both AB antigen, and O blood group neither contains A antigen nor B antigen. Now on the basis of that, the antibodies will differ. So when there is A blood group, the antibodies will be anti-B. When there is B blood group, the antibodies will be anti-A. When there is presence of AB blood group, there will be no antigen present. And in case of O blood group, the antibodies will be A and B, that is, both anti-A and anti-B antibodies will be present there. So how can we predict the ABO blood group of a child? Now we can take a condition where a male having blood group A, which is homozygous in nature, that is, one capital IA with capital IA, which is homozygous in nature, is crossed with a female having blood group B, which is heterozygous in nature, that is capital B with small I, which is heterozygous in nature. What will be the blood group of the child? So now from these two things, the presence of the alleles, we can predict the blood type of the child. So if a homozygous male is crossed with a heterozygous female having blood group B, what will be the blood type of the child? Here is a table format given where a homozygous male is crossed with a heterozygous female, and the results of the cross are: in the first cross there will be expression of capital IA and capital IB, which is AB blood group, as both of them are dominant alleles. In the second one, the same result will take place, that is capital IA and capital IB, when as in this case also the expression will be AB, as both of them are dominant allele and they are showing codominance. When the cross takes place between small I and IA, the expression will be A blood group, as capital IA is a dominant allele. And this fourth one will also be same, there will be expression of capital A IA, as it is a dominant allele, and the blood group will be A. So in the genotypic ratio, there will be a ratio between heterozygous AB and heterozygous A, and the ratio will be 1:1, as there are two AB blood groups and two A blood group, and the ratio will be 1:1, which shows that the baby can have a genotypic ratio of 50-50, and there is a 50-50 chance of having a heterozygous AB and a heterozygous A blood group. And in the phenotypic ratio of AB is to A, there will be a 50-50 chance of having AB blood group and A blood group. So from the previous way, we can predict the blood group of a child if we know the blood group of the parent generation based on alleles. Based on alleles. What is meant by based on alleles? If we know that if the parent is either homozygous or heterozygous, or either homozygous or both of them are homozygous, or both of them are heterozygous, certain kind of combinations can take place if we know the type of alleles present in the parent generation, then we can predict the blood group of the child. So according to the 4 different type of maternal and paternal blood groups, there will be 4 into 4, 16 different combinations which can be used to predict what can happen or what kind of blood group the child can have. Here is a table format given where all the 16 combinations of the predicting ABO blood group of children has been given. So now here are AA, which resembles A blood group, BB, which resembles B blood group, OO, which resembles O blood group, AO, which resembles A blood group as O is recessive in nature and A is dominant in nature. There will be BO, which is also B blood group as B is dominant and O is recessive, and there will be AB, where A and B both are expressed as they are co-dominant in nature. Here is the male gamete, here is the female gamete. On the basis of that, the cross will take place and the results will be observed if we do all the 16 kind of combinations present in the parent generation. Hope this video will help you, and for any kind of query or information regarding this video, you can comment in the comment box. Thank you.